Hello everyone, I'm Dr. Sahithi and today we are going to deal about fertilization. So, what do you know about fertilization? It is the process of fusion of male and female gamete, that is sperm and ovum. So, in this class in detail we are going to deal about how the fertilization occurs. Coming to the definition of fertilization. So, fertilization is the process of fusion of two highly specialized, mature, haploid germ cells, that is, ovum and sperm, resulting in formation of most undifferentiated diploid zygote. So, the fusion of specialized and mature, the germ cells are always haploid in number. So, the process of fusion of the specialized mature haploid germ cells, which results in the formation of diploid zygote, which is undifferentiated. So, this is the definition of fertilization. The nucleus of ovum becomes the female pronucleus and the head of the spermatozoon or sperm separates from the middle piece and tail and transforms itself into the male pronucleus. So, during the fertilization, the spermatozoa or the sperm gets separates, the head gets separates from the middle piece and tail, and the head forms the pronucleus, and the nucleus of the ovum forms the female pronucleus. And the fertilization is antithesis of meiotic cell division after the fusion. These undergo changes like those in a typical meiotic division, leading to the formation of an embryo having two cells. And some of them say that there is no one cell stage. So, what the word here antithesis refers to? So, as we all know, in meiotic division, the haploid cell is going to give haploid daughter cells. But whereas in fertilization, what happens? It is opposite to the meiotic division. The antithesis word refers to the opposite. So, fertilization is opposite to that of meiotic cell division. Whereas here, two haploid cells are going to fuse to form diploid zygote. So, where does the fertilization occurs? It occurs in the ampulla, that is the lateral 1/3 of the uterine tube. So, what is lateral? The one which is away from the midline. So, if you consider this is the midline and this is the uterine tube, the lateral 1/3. So, this part is known as the ampulla, and here the fertilization occurs. So, let us see the main three stages of fertilization. So, what is important in fertilization? Mainly, first point, approximation of gametes. So, the gametes are formed. In male, the male gamete is formed, and in female, the female gamete is formed. So, the approximation of both the gamete is important for the first. Next, after approximation, contact and fusion of gametes is important. After that, the effects or the results of the fertilization will be seen. So, in approximation of gametes, we will see about the male gamete, how it is going to occur, and female gamete. So, for male gamete, that is for sperm, first the spermatozoa transport from the seminiferous tubules to the epididymis to undergo maturity and motility of the sperm. And later from there it is transferred, and from there we can see the transport of the sperms. And later the fate of the sperm in the female genital tract is very important. And after reaching female genital tract, it undergoes capacitation. So, these are the points which come under the approximation of gametes of male gamete. So, in female gamete, as we already studied about the structure of ovum, that is very important to know, and how it is transported to the uterine tube, and what is the viability period. So, first let us see about the male gamete. In male gamete, what we have read first? The spermatozoa. So, this is the steps of transport of the spermatozoa. One, from the testis to epididymis. From epididymis through vas deferens, it enters the seminal vesicles, and after seminal vesicles, it passes through the prostate, and then through urethra, it is ejaculated out. So in testes, in seminiferous tubules, the sperm are produced. And coming to the two, that is epididymis, they are stored and matured in the epididymis, the sperm. And the three, that is vas deferens. From the epididymis, it carries the sperm to the seminal vesicles. Coming to the fourth, in seminal vesicles, the secretions which are rich in fructose is secreted, which provides the energy to the sperm. So, as the sperm passes through the seminal vesicles, it produces some secretions which are rich in fructose, which is a source of energy for the sperm. Next, secretions of prostate gland. So, as after seminal vesicles, it passes through the prostate gland, and here the secretions are rich in ions, citric acid, and acid phosphatase, and fibrinogen, which is a clotting factor, which is very important, and it clots the semen. When it clots the semen in the female genital tract. So, these are the secretions of the prostate gland. After prostate gland, the secretions of bulbourethral glands act as an pre-ejaculatory lubricant. So, in this picture, it is not there, but bulbourethral glands are present below the prostate gland, and from here, its secretions help as an pre-ejaculatory lubricants. So, these are the steps for the transport of the semen or the sperm. So, we have seen how the semen is going to transport it. So, now how much quantity of semen is ejaculated? 2 to 3 ml of semen is ejaculated, and the pH of it is 7.2 to 7.6. So, what are the contents of the semen? Mainly the seminal vesicle secretions, which is present about 60%, and prostate secretions, which is about 25%, and bulbourethral gland secretions, which is about 5%, and the spermatozoa or the sperm is about 10%. So, this forms the semen, and the count of the sperm in semen is 100 million per milliliter. So, now we have seen the transport of the sperm. So, it is transferred to the female genital tract after ejaculation. So, we will see the fate of spermatozoa in the female genital tract. So, the viability period of spermatozoa after ejaculation is 24 to 48 hours. That is 1 to 2 days. And in 200 to 500 million sperms, about only 300 to 500 spermatozoa reach the site of fertilization. What is the site of fertilization? Ampulla. And prostaglandins, which are present in the semen, stimulate peristaltic contractions of the female genital tract, and movement of their tails through the uterus and tubes assisted by the muscle contraction. So, what happens is, after ejaculation, the prostaglandins which are present in the semen will stimulate the muscle contraction of the female genital tract, and the muscle contraction of this female genital tract will help in the motility of the sperm to the uterine tube. Next, let us see the pH, how it affects the sperm motility. So, the vaginal pH is acidic, then it decreases the sperm motility. Whereas, if it is alkaline in nature, it increases the sperm motility. So, this is the fate of sperm in the female genital tract. The main important final process of maturation of sperm in the female genital tract is known as capacitation, and it is species-specific interaction between the sperm and ovum, and it takes about 7 hours of duration after ejaculation, and it involves changes in sperm cell membrane and signal transduction, which includes like calcium influx, which helps in the increased motility of the sperm. And there is an immunological reaction which occurs between the glycoprotein, which is fertilizin, which is present on the ovum, and the anti-fertilization glycoprotein which is present on the spermatozoa. So this is species specific, and the main immunological reaction occurs between the fertilizing of the ovum and anti-fertilizing of the sperm. So these are the three steps which we have seen in the male, that is the transport of sperm and its fate in the female genital tract and the capacitation of the sperm. So now let us see about the female gamete. Structure of ovum at ovulation. As we all know, the secondary oocyte, which contains of haploid number of chromosomes, that is 23 chromosomes, which is enclosed in from inside to outside, vitelline membrane, zona pellucida and corona radiata. And next, coming to the transport of ovum. The fimbriae of uterine tube moves over the ovary at the time of ovulation, and the ciliary beads of fimbriae sweeps the ovum into the infundibulum. So here in the picture you can see, right, the oocyte is released. So these are the fimbriae of the uterine tube. So during ovulation, what happens? The fallopian tube fimbriae will move over the ovaries to sweep, like to get the ovum into the uterine tube. It occurs, and the ciliary bead which occurs which helps in the transport of the ovum, and where it will reach at the site of fertilization, that is ampulla. And the ciliary beads of the uterine epithelium and the muscular contractions of the uterine tube are responsible for the migration of ovum. So both the ciliary bead, the epithelium which is present in the uterine tube, also will form a wavy movement which helps in the movement, like sweeps the ovum from the ovary to the ampulla, and also there is continuous muscular contraction occurs in the uterine tube which helps in the migration, and the viability period is about 24 hours. So first we have read about the approximation of gametes. Now the approximation of gamete is done. So next we are going to see about the contact and fusion of gametes, and it mainly involves 4 processes, that is acrosome reaction, disintegration of barrier, and the calcium wave in oocyte cytoplasm, and last but not least the nuclear fusion. So first let us see acrosome reaction. Acrosome reaction occur when sperm comes in contact with corona radiata of ovum. So which is the last layer of the ovum which is surrounding the ovum. When the sperm comes in contact with the corona radiata, the acrosomal reaction occurs, and perforation develops in the acrosome. So you can see this is the acrosome, right? And here you can see the perforation. Through this perforation, the acrosomal enzymes are released out. And what are the acrosomal enzymes? Hyaluronidase, protease enzyme, that is acrosin, and acid phosphatase. These are released from the acrosome. So now the enzymes are released. So what happens next? Next, the disintegration of the barrier occur. So first, the outer layer, that is corona radiata. The penetration of the first barrier on the release of hyaluronidase. So as we all read in the structure of ovum, the last layer, the outermost layer, corona radiata, the cells are held by a matrix hyaluronic acid. So to digest that in the corona radiata, hyaluronidase is released from the acrosome of sperm. Next, coming to the zona pellucida, we have here the glycoproteins to which the head of the sperm gets binded, that is ZP2 and ZP3 of the zona pellucida of the ovum, whereas the acrosome acrosin, which is released from the acrosome, causes the digestion of the zona pellucida. And coming to the vitelline membrane, the integrin peptide of the vitelline membrane is attacked by the disintegrin peptide of the sperm and initiates fusion. So the corona radiata, which is digested by the hyaluronidase, which is released from the acrosomal enzymes, and the zona pellucida, when the sperm head get fused to the ZP3 of the zona pellucida, the acrosin will act and help in disintegration of the zona pellucida. And coming to the vitelline membrane, the vitelline membrane contains of the peptide known as integrin, to which the disintegrin peptide of the sperm will be attached, and it acts and helps in the fusion. So, in this way, the disintegration of the barriers occur. Next important thing is calcium wave in the oocyte. So, what happens? The contact of sperm with the vitelline membrane of oocyte triggers the calcium wave in the oocyte cytoplasm. What does this calcium wave in oocyte refers to? It refers to the increased number of calcium ions in the cytoplasm of the oocyte. And what does it do? It stimulates the meiotic division of the oocyte. As we all know, in ovum or the secondary oocyte, it will be stopped in the second phase of the meiotic division. So, as soon as the sperm head will get fused with the vitelline membrane, it complains by the calcium wave in the calcium wave in the oocyte. It helps in the completion of meiotic division. And another thing which occurs is zona reaction. What is zona reaction? The contents of cortical granules released in the pre-vitelline space and induce modification of ZP2 and ZP3 and prevent polyspermia. So here you can see, right, the sperm and the egg go under fusion. It crosses the vitelline membrane. Immediately what happens? In the periphery of the cytoplasm, as we read, we will see cortical granules. So these cortical granules are releasing enzymes into the pre-vitelline space, and this process is known as cortical reaction. So what happens by cortical reaction? These enzymes which are produced, which induce modification in the ZP2 and ZP3 of the zona pellucida and prevent the entry of another sperm, which indirectly help in preventing polyspermia. So these are the two important factors which will be done by the calcium wave which occurs in the oocyte. Nothing but it stimulates the meiotic division and the zona reaction. Don't get confused between the cortical reaction and the zona reaction. Cortical reaction is nothing but the cortical granules which releasing the enzymes into the pre-vitelline space, whereas the zona reaction is nothing but the release of the enzymes which cause modification in the zona pellucida and prevent polyspermia. So these comes under calcium wave oocyte. Now coming to the nuclear fusion. The approximation of pronuclease takes place near in the middle of the cytoplasm of the ovum. And during entry, the sperm head makes an rotation of 180 degrees within the oocyte cytoplasm. And here you can see, right, as soon as the sperm enters, the pronucleus get attached. Here also you can see this pink one is the female pronucleus and this yellowish white one is the male pronucleus, which undergo fusion and the rest process occurs. What happens? Firstly, the replication of DNA takes place to form a second chromatid and two centrioles appear here. And after that, the nuclear membrane disappears of both male and female pronucleus, and the spindle fibers are formed between the centrioles, and the chromosomes from each pronucleus, that is from the male chromosomes and the female chromosomes, organize on the equator. And after that, one chromosome of each pair move to one end. So half of it move to the one end and other half move to the another end, and which helps in the formation of two cells having 46 chromosomes, that is diploid in number. So this process is similar to that of mitotic division. So in this way, after nuclear fusion, the diploid number of zygote is formed. So these are the steps which we have read in the approximation of the gamete, and also the contact and fusion of the gametes. Now, after contact and fusion, we have seen the diploid number of zygote is formed. And what are the effects and results of fertilization? Firstly, after when the sperm head enters the vitelline membrane, it completes the second meiotic division. And also the restoration of diploid number of chromosomes occurs, as the gametes both cells are only haploid in number, but by fusion we will get diploid number of chromosomes. And the determination of chromosomal sex of the future individual also occurs here, depending upon the X chromosome and the Y chromosomes of the male gamete. And next, coming with the initiation of cleavage of zygote, which will be dealt in the next class in detail. And the determination of polarity and bilateral symmetry of the embryo. So here the polarity and bilateral symmetry of embryo is nothing but the cells undergo like specialization to form a particular tissue, which occurs after fertilization. And next point is genetic diversity. So the mutation which occurs and also the gene which is carried in the population all comes under the genetic diversity. So these are the results of the fertilization. And one thing too important is the embryo contains only maternal mitochondria, whereas the sperm mitochondria will be discarded before. Next, coming to the factors which are important for the fertilization. So we have seen approximation of the gametes, contact and fusion of gametes, and after contact and fusion, how the results of fertilization will be seen, we have seen. So for fertilization, what and all factors are important? The normal functioning of reproductive organ, obvious, as to form a mature haploid number of germ cells, both in the male gamete and the female gamete. The proper functioning of reproductive organs is very important. And also another process which is known as chemotaxis, which is nothing but when the sperm enters the female genital tract, and when the ovum will be in the uterine tube, there is a process which occurs which helps the sperm to move towards the ovum and which helps in the fertilization. This process is very much important. Next, coming to the coordination of time interval between insemination and ovulation, and that is the main factor which to be considered here is the viability period, as the viability period of ovum is just 24 hours, whereas the sperm is 24 to 48 hours. So these two factors are very much important for fertilization. Next, the number of sperms reaching the fallopian tube is also important, and the other factors of semen which influence the fertilization. That is nothing, we can take it as the enzymes like hyaluronidase, acrosin, and all, and prostaglandin, which helps in the motility of the sperm. So these and all factors are very much important for the fertilization. So in the next slide, I have given a small activity, nothing but the match the followings. So here I have given the enzymes of like during acrosomal reaction, what are the enzymes which will be released and how it is going to disintegrate the layers of the ovum. So I have given hyaluronic acid, acrosin, and disintegrant peptide of the sperm. So in which way it will disintegrate? The hyaluronic acid will disintegrate the outermost layer, that is the corona radiata, whereas the acrosin enzyme which disintegrate the ZP2 and ZP3, mainly the glycoproteins of the zona pellucida, and the disintegrant peptide which gets attached with the integrant peptide of the vitelline membrane and helps in the fusion. So the important questions which can be asked in the topic of fertilization can be the stages of fertilization, which I have mentioned in the first slide, and you can like make a flow chart of it, and for easy understanding, and also if you present in form of flow chart, you can score good marks actually. And you can also like in short time you can give more information. And the definition of fertilization, which is very important, and they can also ask explain fertilization in detail. Thank you.